a precursor to clinical disease. And our current knowledge pertaining to colonization, the risk factors for colonization and the risk factors for progressing to symptomatic C. difficile really is based in the adult literature. So up here in the top left, kind of summarizing what we know about risk factors for colonization in the adult population. Recent hospitalization, chronic conditions such as dialysis, exposure to corticosteroid or immunosuppressants, gastric acid suppression, and kind of in a protective sense if someone has antibodies against certain toxins. The pediatric review from which I obtained that image on the slide before, summarized a number of case control studies on the pediatric side, but the results were quite conflicting in terms of whether the impact of antibiotics or prior medical conditions really played a role in colonization. And then on the bottom left here, in terms of risk factors for progressing to symptomatic CDI, there was a recent meta-analysis that looked at what factors might predispose patients to developing symptoms. Really, I think most authors feel that the gut microbiota plays a very important role because the presence of your diverse microbiota can, prevent C. difficile from kind of growing and reproducing, because the other microbiota can produce direct resistance mechanisms. They can use nutrients that the C. difficile needs. And there's also host antibody mediated immune responses that probably mediate this process. And so, we don't really know what is the natural history of C. difficile colonization among pediatrics, let alone among the pediatric oncology population. So during my master's thesis studies with Dominic, I wanted to get a better understanding of this. And we conducted a scoping review to identify the studies that reported on the prevalence of colonization, risk factors for colonization, and the natural history. We used a comprehensive and reproducible search strategy like one would use for a systematic review, and we identified 6 observational studies. And these took place as single center studies and were conducted over the course of a few decades. All of them, or almost all of them, were prospective in nature. 5 of them looked at the inpatient setting and 1 looked at the outpatient setting. In terms of the included patients, the spectrum was quite broad. They really ranged in age in terms of which children were included, as well as their underlying oncologic diagnosis. For the most part, all of them used fresh stool to detect C. difficile using specific stool culture medium, but toxin detection methods varied significantly. So some studies used cytopathic effect detection, enzyme-linked immunosorbent assays, or EIAs. Some of them used PCRs, and one study actually just used PCR alone for detecting the toxin B gene. And so most of the studies were actually able to differentiate between the proportion of toxigenic and non-toxigenic strains. And so overall, when we pulled the results, 365 individuals were tested among these 6 studies, and 86 were found to be colonized. And so this was a prevalence of roughly 20 to 25%, but this ranged about 4-fold over the different studies, from under 10% to almost 40%. Toxigenic strains were differentiated, as I said, in several of those studies as well. In terms of risk factors for colonization, 4 studies looked at risk factor identification, but the results were all conflicting. And the problems were, the studies were small in size. Some looked at colonization at admission versus during admission. Different age groups were involved. And so it was very difficult to find a consistent and significant relationship between known factors from the adult side, such as antibiotic exposure and healthcare exposure. And then lastly, there was one study that looked at the natural history of patients who were colonized by following them on a weekly basis. And there were 2 studies that looked at how long or how frequently C. difficile was detected in someone's stools after they were diagnosed with CDI. But again, these are very small numbers. In the study that looked at weekly surveillance of patients in the outpatient setting, they found 4 who were colonized, but none of them went on to develop symptomatic disease. And then for the other 2 studies that looked at detecting C. difficile after someone recovered from C. difficile infection, they actually didn't clearly differentiate between those who were symptomatic and asymptomatic. So in conclusion, we couldn't really identify or properly describe the natural history of C. difficile colonization in this patient population. So, very briefly, just sharing some other results from our experience, we conducted a retrospective cohort study at McMaster Children's on our oncology ward. This took place over an 18-month period, and we looked at the development of C. difficile infection up until May 31st, 2018. And so we included patients who were over the age of 1 and under the age of 18 who were admitted to the oncology ward and had a diagnosis, a primary diagnosis of cancer. And we included those who had screening swabs for MRSA or VRE that were done within 72 hours of hospital admission per kind of our hospital policies. And we tested these swabs for the presence of C. difficile, specifically toxigenic C. difficile, using a loop-mediated isothermal amplification test. This is a PCR test that we use on the clinical side when someone has symptomatic disease and we're looking for C. difficile. And so we collected swabs from 74 unique patients. The median age was just under 7 years, and the majority were male. Similar to kind of the general oncologic population on the ped side, the majority had hematologic malignancies. 9 patients had a previous episode of CDI. And we collected a total of 259 swabs where we had C. difficile colonization data available. So a median of 5 admissions per patient during the study period. Unfortunately, there were a lot of admissions where we didn't have C. difficile colonization data. And so, roughly 50% of these instances were due to a swab not making it to the research lab, and the other 50% were missed opportunities where swabs weren't collected properly. So it obviously did impact our findings. And so graphically, these were kind of all the patients that the unique patients that we had in our study. Each line represents a unique patient. Each orange bar represents the duration of a hospital admission. If there is a orange open circle, this represented someone who tested negative and didn't have colonization. And if there is a kind of closed blue dot, this represented identification of C. difficile, and in our instance, it represented asymptomatic colonization. And as I mentioned, there were limitations to kind of the results we could generate because of the number of swabs that unfortunately weren't collected and tested. So we weren't able to identify any demographic or clinical features that were predictive for colonization. And then looking more closely, we had 9 patients who were found to be colonized on at least 1 admission. And again, their baseline characteristics were not significantly different from uncolonized patients. And when we did a regression analysis looking at these repeated measures or repeated assessments, we also couldn't find either demographic or clinical features that would predict, which admissions these patients would be found to have colonization. None of the 9 patients went on to develop C. difficile. As I mentioned, or as you can see here in the graphic, there are 2 green dots for patient 2 and patient 4. And these patients had C. difficile prior to the first positive colonization swab. And in one instance, patient 2, we found that within a week, they no longer had C. difficile identified. And then for patient 4, their next swab was 30 days later, and they had a positive colonization identified. patients 2 and patients 8, you can see had a number of blue dots, so they were found to be colonized on numerous admissions. And from these 2 patients, we found that colonization either lasted from less than 7 days or possibly up to almost 2 months, depending on which patient we were looking at. So all that background, just to lead us to a case. This is a scenario that is not that uncommonly encountered on our oncology ward. So this is a 16-year-old girl with CNS germinoma. She was diagnosed in October of 2019 when she presented with shock and was found to have panhypopituitarism and adrenal insufficiency. Now that she's kind of been with us a little bit longer, her past medical history list is a bit longer there. So she initially had ADHD, known ADHD, and then subsequently with this diagnosis was found to have central diabetes insipidus, panhypopit, and she ultimately required a G-tube insertion for nutritional support. Her medications include DDAVP, levothyroxine, omeprazole, hydrocortisone, vitamin D, and estradiol patch, as well as growth hormone injection. And so during her hospital stay, she develops fever while she's neutropenic, and piperacillin tazobactam is started for empiric coverage. This is commonly what's done for our patients who have neutropenia and fever. But unfortunately, she subsequently develops loose stools, and her samples are sent to the lab for testing. And this is the result that's reported. So C. difficile toxin gene is identified by our LAMP assay. And so a decision to treat is made by the primary oncology team. The problem is this happens actually 6 times over the course of less than a year. So in December 2019, she receives a course of oral metronidazole. Again, less than a month later, she has symptoms and she tests positive and she receives oral metronidazole again. And then subsequently in April, May, twice in May, and in June as well. I mean, all of these episodes are separated by, less than a month in the later part of 2020, and she receives an oral course of vancomycin each time. And then on the last episode, because she's had so many episodes, a decision is made to develop a tapering schedule for her. And these are the kind of questions that we run into when we are asked about a case like this. Is it true symptomatic C. difficile infection each time? Because sometimes the abnormal stools occur while she's on chemotherapy. Sometimes the abnormal stools occur while she's on antibiotics. So she clearly has other causes for her symptoms. And sometimes the abnormal stools are tested even before she meets that threshold of 3 abnormal stools in a 24-hour period. And then so the next day, her symptoms are significantly better. And the question is does she truly have CDI? Does she not? Should we just treat because she's possibly shedding? And sometimes the stools go back to normal within 1 day after treatment is initiated. So there are a lot of questions that surround whether this is true infection each time, and this impacts kind of decisions about whether she should be treated and what she should be treated with. And so just to kind of wrap up this portion before I hand it over to Yingfu, diagnosing C. difficile infection is problematic. And current tests can look at 3 different questions. So first of all, there are some tests that can identify if C. difficile is even present. So there are specific stool culture media that are available. The sensitivity is high, but the specificity is moderate or variable. The problem is this is not of great clinical use, especially if you're just looking for any C. difficile, regardless of whether it has the ability to produce toxin or not. Subsequently, there has been a move to GDH-based tests, and again, looking for the presence of C. difficile. The problem being that even though it's very sensitive, it's not very specific, and so these tests often need a confirmatory test to follow. However, these tests are widely available, which is ideal. The next group of tests kind of answer the question, does the C. difficile that we identify have the capacity to produce toxin? So again, you can use a culture-based method, and we're looking for toxigenic culture-based methods here, where not only are you identifying C. difficile, but then you're taking those isolates and then testing them using C. difficile cytotoxin neutralization assays, and you're looking for cytopathic effects. So there is better specificity here. It maintains its high sensitivity, but the availability is limited because of the culture media as well as the neutralization assay media. And so these are often reserved for reference labs and epidemiological studies and less so for clinical use. Then again, more recently, PCR-based tests have come about that are looking for the toxin gene, such as nucleic acid amplification tests. And these are very sensitive tests, but maybe not as specific. The availability is increasing as PCR-based technologies are more widely available. I think the startup cost can be pretty high because you're purchasing new machinery, but once you have those processes in place, the cost of the tests and the media to actually run the tests, on a per test basis is not as expensive. The clinical problem is these tests can provide a lot of false positives. Perhaps what we're running into in the patient that I described. And then lastly, some tests look for the presence of C. difficile toxin itself, so not the gene, but the actual toxin. Cytotoxin assays are available with high sensitivity and specificity, but again, these can be more costly and with limited availability. And then on the flip side, enzyme immunoassays were thought to be a very pragmatic approach to diagnosing clinical disease because these assays are more widely available, but the sensitivity can be quite poor. You have to detect both toxin A and B, and so the use has been quite limited. So some reference labs will use a two-tier approach, for example, screening with an antigen-based test like GDH and then confirming with a toxin assay, a nucleic acid amplification test-based assay, and some reference labs will go straight to a PCR-based test for the toxin gene. So kind of going back to her case, this young lady was actually hospitalized the entire time from October to the end of July. She had finished her chemotherapy and radiation therapy, and her stay was prolonged because she had issues with her diabetes insipidus. And then, unfortunately, 2 weeks later, when I was on service, she presented to the emergency department with a one-day history of fever and diarrhea. And upon arrival, she was tachycardic, very hypotensive, requiring aggressive fluid resuscitation and inotropic support with almost maximal doses of norepinephrine and epinephrine. She's chronically leukopenic, and so broad-spectrum antibiotics were started. The PICU was consulted to see her, and they were admitting her, and they asked us about empiric coverage for C. difficile. Is this necessary given how many supposed episodes she's had? And then the next day, her blood culture is positive for gram-positive bacilli. So there was a lot of concern about her clinical status. But there were so many conundrums in this case, kind of built up because of these episodes over her admission where she had symptoms and she also had positive C. difficile tests. And the questions that we were thinking about were, in this patient who has, this episode of diarrhea, she has fever, she's hemodynamically unstable. Is this CDI? Is this severe CDI? And because of the inotropic support that she's receiving, are we even able to optimize enteral treatment? Because the PICU team was so concerned about the ability to give her even medications enterally with those inotropes on board. So there are a lot of unanswered questions. The limited literature suggests colonization rates can range up to about 40%, and we found a prevalence during our short study period of just over 10%. It still remains unclear which patients and epidemiological characteristics are predictors for colonization. The natural history of colonization, including the duration and progression to CDI, remains unclear. And of course, as we kind of highlighted at the end here, the available tests for C. difficile detection are imperfect. And so at this point, there may be questions and discussion, but I'd also like to hand it over to Yingfu.
Thank you so much, Jacqueline. That was really interesting. I think what we'll do is have Yingfu speak, and then we'll combine the questions at the end, if that's okay.
Absolutely.
Perfect. Okay, so if you stop sharing your screen, then Yingfu can do his. Thanks.
Wonderful. So you get mine to work. Starting from here. Well, thank you so much, Laurie, for the invitation and for the introduction, and Jacqueline for the wonderful introduction on C. difficile. I already learned a lot and have some ideas I want to talk to you later. So my lab has been working on really what we call functional nucleic acids that they can be used for medical diagnosis. And specifically in this case, we're interested in Clostridium difficile. So I just checked CDC USA yesterday, and I screenshot the information shown here. CDI is a serious problem, as you can see, although in recent years, the cases are coming down. So in the latest number from CDC is 2017. There are about 250,000 cases hospitalized in US, and estimated deaths is over 12,000. Of course, costs a lot of money. And so I think Jacqueline has already done a great job on the C. difficile, so I'm not going to talk too much about these bacteria, and it's also the CDI. And one of the biggest problem or the risk for these diseases increase with the patients with antibiotic use. So that's obviously it's a common issue for CDI. And Jacqueline has also mentioned about a few diagnostic tests that are currently used in clinical labs. And the information I've written on top there is also taken from CDC. In general, they can be grouped into 3 types of tests. One is a culture-based, and it's quite accurate. It's a different form of culture-based assays, but the disadvantage is time-consuming. It's the laboratories, right? And the other one is antibody-based test. They're pretty rapid, but limited sensitivity and or specificity. And the other one is amplification-based nucleic acid targeted test, either PCR, LAMP, or we're using a different techniques, which are sensitive, but often it come with high number of false positive. This is the so-called, over-diagnosis. To my knowledge, there are a lot of actually specific strains or epidemic strains like LAP1. Currently, there's really no good clinical diagnostic methods. And so all these methods are listed or used clinically right now are not suitable for identify specific strains that might be more antibiotic resistant, or they may express more toxins or may require different treatment methods. We feel that my lab can come in and maybe make some contributions here, since my lab has been really working on functional nucleic acids and specifically DNAzymes or DNA-based enzymes. Specifically here is that we want to create biomarker responsive catalytic DNA molecules. These are synthetic and made in test tube. They can chemically synthesize, so they can be cheap and widely available. Similar to that is like a DNA version of antibody, or we call DNA Aptamers. Those are biomarker binding DNA sequences, specifically here for C. difficile, then we would target specific proteins for detection. As an interest for me and a group of scientists and engineers at McMaster, we are very much interested in creating simple, cost-effective devices, so like a paper-based analytical device or pad, which include the lateral flow devices and litmus tests, as we often know that when we're in high school, we use these kind of tests. And so they are cheap, right? So it's easy to use, and they produce a color. And but most of these tests, are for simple things like a pH or pregnancy test for hormones and so on. So there's still fairly limited availability for these type of tests on the market. So we thought that we would like to do something like this. And it's also, Jacqueline mentioned about the sensitivity, and the most sensitive test is involved amplification. Amplification mostly done by PCR, but this LAMP also become very, very popular. But lamps still need a machine and need a specific temperature. So we're kind of interested in using a technique, which I write down here. In the middle is the isothermal nucleic acid amplification method. So we have developed multiple methods, but the ones we are actually very much interested in is called running circle amplification, or RCA, which I will talk a little bit later in our sensor development. Now, so we're making synthetic DNA molecules that have catalytic or binding or both functions. And how will we do that? So nowadays we can easily produce synthetic DNA libraries. And if you spend $500, I can give you a library that contain really gazillion sequences. In fact, we can make a library now that contain all the way to 10 to the power of 18 different sequences. And from within these sequences, we believe there are some magic molecules that we can isolate. So we start, perhaps you can see my mouse here, we call that combinatorial library. So they contain many variations of the sequence. Then we are using a technique that we can isolate the ones that we're interested. I call it active molecules. In the process, we throw away the inactive ones. And we use simple things like gel electrophoresis or using affinity binding technique, right? So if we can partition these two group of molecules in this step, then we amplify the active ones using PCR. Then we generate a second pool, and then we simply repeat this cycle many, many times. So this technique has been invented by Jack Shostak and Larry Gold many, many years ago. So these are the techniques that we have been using in the lab for many, many years. So I will quickly tell you three projects we have now published for different reasons. So the story one is really going after, we're going after a strain-specific DNAzyme that can detect epidemic C. difficile strain. And the second one, we call the antigen test, and Jacqueline mentioned about that already. So the antibody-based test for GDH. So we're thinking that we can do a DNA aptamer to replace antibody. Then we can detect GDH too. So that will allow us to detect any C. difficile strain. So that GDH is specific to any C. difficile. Then the other one is we've been working on toxin B binding aptamer, because not all C. difficile strains are actually infectious, and some of that does not produce toxin. So there's really not a problem there. And but for infectious strain, they express either toxin A or toxin B. Actually, all the strains express toxin B. So we saw that if we can develop a sensor that can recognize toxin B, then we'll be able to tell you whether, so if we have a device with all these three type of sensors, then we'll be able to tell you whether it's a C. difficile, whether it's a toxin B containing C. difficile, or whether it's actually a specific strain of C. difficile that need a different treatment. So first, strain-specific DNAzyme. We want to detect, actually this this work was done in collaboration with Clostridium Li. I believe now she is in Victoria, but when she was here, I got linked to him through Jerry Wright when I just started getting interested into CDI. And And she said, the current lead right now is to identify specific strains of C. difficile. And she gave us a strain called the BI027H. H stands for isolated in Hamilton. And she wanted us to see if we can get a probe that recognize this. But at that time, we have no idea where to start because we do not know a specific biomarker for it. But fortunately, we already developed a method that we do not require a biomarker to get a specific sensor for a bacteria. And this is I write down here as RNA-cleaving DNAzyme. So these are the DNA molecule that contain, everything is DNA, but in that sequence all contain RNA and illustrated here. So RNA, DNA differences at these 2' position. So here, RNA has a 2' hydroxyl, which can be used as nucleophile to attack this phosphodiesterase bond and then cleave it. So you generate 2 fragments. So we call this RNA-cleaving DNAzymes. And so I already told you that we can do selection. So we basically take a library and we add these RNA-containing substrate to each member of the library. And then we ask the molecule that can cleave in the presence of a specific strain of C. difficile, then do the reaction. Then we isolate this broken down product, right at the linkage of RNA. So isolate these pieces, we amplify, then we get double-strand DNA, and we convert them into single-strand DNA. We repeat this cycle again. Now, if you notice, I have an F and Q here. These are the 2 nucleotides we modify, one with fluorophore, the other one with a quencher. So of cleavage, and these will generate a signal because the quencher is no longer associated with this molecule. So you can simply detect fluorescence. They can give you a real-time reporter if you actually have the enzyme. So in this case, I said, we don't know the specific marker. So we basically just take the whole mixture from the cell, then we mix with the library, and the one that can binds and cause the cleavage, and we'll isolate them in this step, okay? And to engineer the specificity, we do a step called counter selection. So imagine in the library, we have 3 types of molecules. One, they have cross activity with other bacteria. Okay, we throw them away. And then also the one that does not cleave, catalytic inactive, they will not get selected because we only isolate these small pieces, right? So if they don't cleave, this longer piece and won't get isolated. And the ones we really want is this green molecule. So that can be isolated through 2 steps. One, it does not cleave in the counter selection step with other bacteria, but cleave in the positive selection step. Now they're getting shorter and we isolate these pieces in the positive selection. So that's the method we use very often in our laboratories, okay? So we started with E. coli test. So we're basically using Bacillus as a counter selection target and E. coli as a positive selection target. And these 2, Ali and Sergio, are now long gone. Sergio actually has his own company now doing different things in Hamilton. And so they did the selection. I'm not going into details. It's a tedious process to generate this probe, but in the end, we do have a probe. They can recognize E. coli, but not Bacillus, okay? So, and this is the sequence. And if we actually do the fluorescent test, you can see E. coli and these other bacteria. And you can clearly see that's quite specific. We went on and got actually many different bacteria from Jerry's, Eric, and Combs' lab. And we show here is E. coli, you had generated fluorescence with other bacteria is actually pretty much not active. So that's a great encouragement. And then we work with Christine, apply this method to isolate the DNAzymes that can now respond to the specific strain of C. difficile. So we did in a counter selection step, we're using E. coli, we're using Bacillus, and also we include C. difficile strain called CD630. It does not contain a toxin or is not infectious. So we basically want to eliminate as much possible the cross activity with non-targeted strains of C. difficile or the other species of bacteria in this step. The ones that don't cleave, then we challenge it with the biomarker from the C. difficile, okay, this strain. And the positive selection step. Now we isolate this, then we go, we amplify. Now this work is done by Dr. Zifa Sen. He's actually running his own lab in China now for many, many years. So he did many rounds of selection. We isolated a whole bunch of sequences written here. And this particular one has the highest activity, as you can see here. Okay, so this is the strong cleavage band. If it cleaves, it generates this cleavage band. Okay, so it's uncleaved and cleaved. So we named this guy called IFD-CD1. And then we have, then we start to do the specificity test. So it's a whole bunch of gram-positive bacterial and gram-negative bacterial. And this is C. difficile, that specific strain. As you can see, the cleavage only happens to this C. difficile. Then we also, over the years, we have accumulated many different strains of C. difficile. And so here's the BR027H, and here's the other strains of C. difficile. As you can see that this probe is highly specific. And the sensitivity, we can actually detect all the way down to 500 cells per mil. And I don't want to go into details here. As I told you that we don't know what's the biomarker to begin with, right? So, but we actually made a huge effort of identify what these DNAzyme respond to. And it's a huge effort. In the end, we find actually this DNAzyme binds to a shortened version of transcription factor called the TCDC protein. Okay, so this is really a major effort done by Zifa. And we also, the data we have not published. So we're also looking at whether these probes are useful to identify these specific strain. So we went to the freezer of Christine's lab and pulled out many different clinical sample, then start to test whether our probe can identify these specific strains from these patients. The red dot each here, maybe I should, I have a simplified version. So H means healthy people who do not have any CDI infection. As you can see, this is very clean, right? So then we test 100 CDI patients, and we know we gene typed two different C. difficile TCDC strains, and through genomic sequencing. And we find our DNAzyme only specifically pick up this called the TCDC24. There are probably about 30 different versions of TCDC gene. And obviously that the DNAzyme we got can detect these specific ones. That's what we initially set up to do. Our work is continuing, and this is actually more of like a lab1 strain. And so currently our work is continuing, trying to isolate DNAzyme for other common strains of C. difficile like lab2 and lab7. So hopefully we'll get a panel of strain-specific DNAzyme that can identify different epidemic strains of C. difficile. Now, this is the previous story is really going after a specific strain of C. difficile. And we have a couple of other stories looking at, so now we can identify the strain and we're using DNA. How about, if we want to create a device, it might be good to actually tell whether this CDI, this is C. difficile to begin with. So then we went back and started to develop aptamers that bind to this antigen glutamate dehydrogenase, or GDH, Jacqueline has mentioned, right? So these are known biomarkers. So this work is done in collaboration with John Brennan and a super postdoc fellow called Menu. He's now actually dean in a university in China, returned to run his own lab there. And so in this case, most of the time when we develop aptamers, we actually using linear DNA sequences. But since we are going to deal with biological sample, we saw that maybe we're using circular DNA, because circular DNA is much more stable. We already know that's the case. So we're just going to take a library, we circularize and create these circles, pool of circles. Then we're using counter selection, a positive selection, to looking for the ones that bind to recombinant version of GDH. And we repeat this cycle many times. And in fact, we pulled out 2 aptamers, okay, and they all binds to GDH, and the data shows here, this is, we labeled the aptamer with isotope, and then we basically titrate the concentration. Then we can actually determine the KD here. We settled down on one aptamer called CAP4, and CAP2 is the same way. And CAP4 and the linear version of that, they have huge differences in binding affinity. And also it's interesting, as you expected, that if you actually incubate this with a stool sample, the circular DNA aptamer is much more stable versus the linear aptamer. And then we also develop an assay, in this case, using a ruling circle amplification. So here we actually put, this is a bead-based assay. We put recombinant GDH on the bead, and we throw the aptamer in the binds. Then we take the stool sample, which contained the native GDH, and it will replace the aptamer. Now, so the aptamer will jump from the beads to this. Then we'll take it to do isothermal amplification called RCA. So RCA is a DNA amplification-based technique. It actually using an enzyme from 529 phage. And these polymerase is unique in the sense you take a primer and copy the sequence of the circular template round by round. So in the end, you'll generate a large single-strand DNA molecule that contain multiple building blocks. And we then convert the GDH detection to the detection of the RCA product. And there are many ways of detecting DNA, including putting a dye in the reaction, so you can actually, through the fluorescence increase, because dye binds to the DNA, now generate, enhance the fluorescence. So here's the binding data. We also applied, and this is in collaboration with a company in Toronto, and they provide the patient sample for us. So negative, the people do not have a CDI, and these are actually people has a CDI. And clearly, you know, they can, they are different. So now we have a test. I have Aptamer and a test that can be used to detect the CDI and detect the GDH from C. difficile. Now, the last story I want to tell you is a recent work we just published this year on. We aim to isolate DNA aptamer for toxin B, and toxin B is expressed by all toxigenic strains of C. difficile. So it is a good biomarker for the infectious strains of C. diff. And we actually started with, this is an interesting story, and we started with selecting aptamers that bind to recombinant toxin B. Toxin B is actually quite a big molecule. So that's probably a little bit of a problem to begin with. And the recombinant toxin B is only represent a domain of it. But we find is actually, we successfully developed aptamer, but failed to bind the native version of toxin B. I call it here like N-toxin B, okay? And particularly when these samples stored for weeks, it turns out, okay, and when the toxin B, which is have intrinsic protease activity, during the storage, they actually break it down like this. And this is the SDS gel we run. So in some ways, the aptamer we selected here will not be compatible with the native one. I think in the stool sample, we suspect this problem is going to happen too. So if you detect toxin B, you have to be able to detect these fragments. And so that's motivated us to now doing the selection against this denatured or degraded native toxin B. So we come up with a method. So we basically incubate the toxin B, the degraded toxin B, put it on the nitrocellulose. So nitrocellulose bind all the proteins. Then we throw our library, we incubate that with this nitrocellulose paper, and we wash, get rid of the ones we don't want, and then we just take the paper and elute and heat it in water and recover the DNA molecules that binds potentially to these fragments of toxin B on the paper. Then we repeat this cycle many, many times. So at the beginning, so we're using this dot blot assay to show that these are round 2 pool, there's no binding. Now round 12, as you can see here, the middle intensity here, that actually shows the binding. So we isolate a whole bunch of aptamers, and we focus on this particular one we call DAP1. And DAP1 had this kind of interesting secondary structure, and we actually done a whole bunch of sequence analysis. We also tested for its specificity. So here's degraded native toxin B. It can bind. It can also bind the fresh toxin B. And we also treat the toxin B using heat, and then cool the toxin B. We find our aptamer can still bind to it. But it does not bind to the recombinant toxin B or toxin A or GDH, okay? And here's just a different, the AMSA binding data, so I don't have time to go into details. And interestingly, this is the recombinant, the aptamer, the binding recombinant toxin B. And although it can bind the native, fresh native toxin B, but it won't be able to bind to the degraded one. And we have done this experiment simply by incubate the fresh toxin B spiked into the stool sample. And as you can see, that binding at the beginning is there. Then progressively decrease, and presumably the toxin B is actually degraded with time. Now this is the new aptamer, and it doesn't matter. It actually binds to the toxin B spike in the stool sample within a day, 5 days, and 10 days. So they give similar binding profile. And we also designed a paper-based sensor. I don't want to go into details, but these sensors essentially we have two. It's kind of an origami type of thing. Okay, so we have sampling zone, and we have detection zone and a control zone. And that will allow us, the control zone always generate a signal. So the test zone, it depends on whether we have the toxin B, will generate different intensity of the color. Okay, so as you can see, this is the control zone on top, and they're consistently with that color. And this is actually the test zone, which is actually have different level of color, different intensity of color, depends on how much toxin B is provided in the sampling zone. Here's just additional data shows you. If we're using quantification, we can detect about, this is lab strain, and these cell counts, and we can detect about 100 lab 1 strains of C. difficile. So in summary, we're still not there completely yet, as we would like to do. But what I just want to point out, we have now developed technologies they can isolate specific DNAzymes and aptamers as recognition elements for C. difficile. And we have ongoing projects for DNAzymes recognizing different strains. So here's the one that bind TC for the Humboldt strain of C. difficile. And this is GDH-binding aptamer, and this is toxin B binding aptamer. We, along the way, we have developed a different form of assays, and here's the beads binding assay, and here's paper-based binding assay. And what we're doing right now is actually we're trying to make the test even simpler. So eventually we want to develop something like this, which we demonstrate with a sensor, a DNAzyme sensor for H. pylori. And as you can see, this is the H. pylori, the stool sample that contain H. pylori, and then we can see the color change with it. The rest is the control, the stool sample that's spiked with the other bacteria. And we really want to get these kind of tests. Now, how this test works is essentially we're using a protein called urease. We link that to the DNA, and so here's one form of solution-based assay, and the paper-based assay is similar. So we have the magnetic beads or any kind of beads that contain our DNAzyme aptamer and tagged with urease. So if you have the biomarker of interest, you will release the DNAzyme along with the urease, and then these can be used to hydrolyze urea, elevate the pH of the solution, then cause the change of dye that put into the solution. So essentially we're detecting the pH change, but the pH change is linked to the urease hydrolysis of urea, and that is linked to the presence of the biomarker of interest. So
Yingfu, I'm sorry to cut you off, but we're running out of time.
Okay, so here's the last slide, and it just shows that my group has. Yeah, thank you very much. I didn't look at the time. Yeah, I'll probably go over.
Thank you very much. If you stop sharing your screen, we can jump quickly to questions. So David has a question. He says that C. diff strains are changing. The prevalence of NAP1 is decreasing, but NAP11 is increasing, especially in pediatric patients. Can your algorithm detect NAP11, or does the assay need to be changed every time a new strain appears?
Yeah, so every time, for me, for the sensor side, then we have to generate a new DNAzyme for it. So basically, it takes a few months to generate that probe, but then the rest are modular. So we can actually develop the assay using the existing assay, but we have to create a new probe for it.
Okay, I have a question for Jacqueline. So these kids that you see that already present with C. diff, is that because they've had antibiotics in the past, or is there some level of natural colonization in the population anyways?
You mean during our study period in terms of the kids that we picked up with colonization? I mean, we didn't look down to the level in terms of like, was this, were they picked up kind of in the midst of their treatment courses, so they would have been exposed to antibiotics, versus was this the very first time they had a swab done? The thing is, because of the screening policy for MRSA and VRE, patients wouldn't be screened unless they've been admitted to hospital before. So that might be the point in time when they might have picked up the colonization, and that previous hospitalization might have involved antibiotics. But interestingly, not in this population, but we get asked about kids who are otherwise healthy, who have significant diarrheal symptoms and get admitted for that, with no apparent exposure to antibiotics and no obvious household contacts that would have your kind of traditional risk factors for C. difficile, who come in with a positive C. difficile test. So I think that reflects the fact that it is in the community. There is an element of community acquired colonization and then infection as well.
And just one follow-up. So that patient that you presented that had repeated courses of vancomycin, I mean, that seems crazy to me that if it's not working, that you would keep repeating the same thing over and over again. Are kids not eligible for FMT?
That's an evolving area. So there are certainly some sites that are looking at FMT for pediatrics. I want to say it's better established and with caveats, but maybe tested more and used more in children, for example, with underlying gastrointestinal conditions like IBD. I feel like that area of research with FMT is a little bit more advanced than our use of it on the pediatric side. As you had mentioned before, I was at SickKids before coming here. And so anecdotally, I remember we have one patient that we follow as an outpatient. This one patient is followed by one staff, and it was a teenager, and they had undergone FMT.
Mm-hmm. And it was successful?
At that time it was. And so they were, when I left SickKids, they had been followed for probably about 6 months only so far.
Okay, cool. Well, I think I'm not seeing any other questions, and just because we're kind of out of time, I want to just say thank you very much to both of you. It was super interesting talks. I'm going to applaud. It's hard to give a talk and not get an audience applause, but this is what you're getting. So thank you again. And thanks for everybody who tuned in. I started recording late, so I'm sorry, Jacqueline, I kind of cut off the beginning of your presentation.
That's okay.
Apparently I'm not the Jackie of all trades that I think I am. But we'll make this recording available for people who were not able to join us this morning. So thank you again, and have a great day.
Yeah, great. Thank you so much.
Right.
Have a good day.
Thanks, everyone. Have a great day.